Okay, welcome everybody to the Chapter 13 video podcast on the lymphatic system and your immune system. We're going to describe the structure and function of important lymphatic vessels, their relationship to blood vessels, and how lymph flows in the body. We'll describe the lymph collecting vessels and ID the structures returning lymph to the venous system and explain lymphedema. We'll define lymphopoiesis and discuss the classes of lymphocytes, their importance and distribution in the body. We'll ID and describe lymphoid tissue and organs and trace the pathway of lymph flow through a lymph node. The next section will deal with nonspecific immunity, where we'll explain how physical barriers and phagocytes play a role in nonspecific defense. We'll explain the significance of inflammation and fever as mechanisms of nonspecific defense and summarize nonspecific defenses. Then in the section on specific immunity, we'll explain how antigens trigger specific defenses and the immune response. And we'll discuss the structure of an antibody and the types of antigens in the body fluids and explain the primary and secondary response to antigen exposure. The lymphatic system is a defense system. Specifically, it's designed to filter the fluids between tissues and check them for the presence of things that would cause disease, which are known as pathogens. And it does this by having a system of vessels that run throughout the body that collect this fluid and funnel it to certain regions where defense cells, white blood cells, will identify and then destroy a potential pathogen. It includes cells, tissues, and organs that defend a body against environmental hazards and internal threats. The cells are known as lymphocytes, and the vessels are called lymphatic vessels or lymphatics. Lymphoid tissue and organs are scattered all over the body. We're generally unaware of them except when we are actively fighting disease. Lymphocytes are primary cells of the lymphatic system that respond to invading pathogens and abnormal body cells such as cancer cells, as well as foreign proteins. Most are produced and stored within lymphoid tissues and organs. They're also made in the bone marrow, specifically the red bone marrow, which in the adult, remember, is going to be found in the ribs, in the os coxa, those are the hip bones, in the sternum, in the flat bones of the skull, the proximal heads of the femur, and so on. They're surrounded by lymph, which is a liquid that resembles interstitial fluid, and it is the second liquid connective tissue in the body, the first being blood. If we look at the layout of the lymphatic system, it's relatively straightforward. You have a series of lymphatic vessels and lymph nodes that are designed to drain regions of the body that are most likely to be exposed to a threat. This is why we find a lot of lymph nodes clustered up near the head and the neck, quite a few lining the digestive and respiratory systems, and quite a few around the urogenital system. We also see extensive lymphatic drainage from the limbs, the arms and the legs, with high collections of lymph nodes in areas behind the knee and near the elbow. You can see here on the diagram the cervical lymph nodes collect and filter the fluid that's collected from the head and the neck. The thoracic and right lymphatic duct represent the areas where the filtered and processed lymph is returned to the circulatory system and then again becomes part of the blood. If we didn't do this, we would rapidly accumulate tissue fluid, causing it to swell. This is a condition known as lymphedema that would impede circulation and would ultimately compromise the function of the organ. The axillary lymph nodes are stationed near the armpit, and they drain regions near the shoulder and the arm, as well as areas near the pectoral region. We also see the cisterna chyli, which is an area where lymph collects as it's drained from the abdominal pelvic region. The inguinal lymph nodes are found near the groin. Other lymphatic tissues to be aware of include the tonsils, which guard the entrance to the digestive and the respiratory system, known as the pharynx. The thymus, which is the site of T cell maturation, it lies in front of the heart and behind the sternum, and is essentially both an immunological and an endocrine organ. The spleen, which lies just under the diaphragm at the tail end of the pancreas and serves primarily to filter pathogens from blood. And then mucosa-associated lymphoid tissue, which is found in the submucosa of the large and small intestine, where it's designed to intercept bacteria before it gets a chance to enter the bloodstream and cause disease. And then you also have the appendix down here in the right lower quadrant of the abdominal pelvic region, a blind-ended sac, which at one point was thought to have no function, but we now know it serves both as an immune organ as well as an area where bacteria that populate the gut can grow and repopulate it in the event that the gut bacteria get wiped out for some reason, such as in taking large amounts of antibiotics. Lymphatic vessels carry lymph from peripheral tissues to the venous system. They begin as lymphatic capillaries that have blind-ended sacs where they terminate. They're found in almost every tissue and organ. They're closely associated with blood capillaries. The interstitial fluid flows into the lymphatic capillaries, and the fluid inside the lymphatic capillaries is called lymph. Now, on average, at any one moment in time, the lymphatic system takes up about 15% of the fluid that leaves the bloodstream to bathe the tissues that require an exchange of nutrients and waste products and oxygen and CO2 with the blood. Most of that fluid gets recovered at the venous end of the capillary bed, but that 15% that gets into the lymphatic system is going to be screened for the presence of pathogens. If we look at a lymphatic vessel, it's very similar in organization to a blood vessel. It has a tunica intima, tunica media, and a tunica externa. The intima is primarily endothelial tissue with valves. The tunica media is smooth muscle, and the tunica externa is connective tissue. And we talked a little bit before about the fact that wherever you find a blood vessel, you generally find a nerve, and you also generally find a lymphatic. And the reason for that is that it maintains the structural integrity of the body. If each of these systems had to run an independent route to get from point A to point B, the body would not be as structurally sound as it actually is. Lymphatic capillaries are lined by endothelial cells that have an incomplete or missing basement membrane, so in that sense they differ from blood capillaries. They also originate as pockets instead of forming continuous tubes. They have larger diameters and they have thinner walls, and they look flattened or irregular in sectional view. So if you were to make a close comparison, they would be a lot more like veins than they would be like arteries. Okay? They share a lot of characteristics with veins. The reason for the incomplete or missing basement membrane is to allow the fluid easy entry into the lymphatic system, and once inside, the difference in pressure locks the fluid in the lymphatic system until it makes it ultimately to a lymph node where it'll be filtered, checked for the presence of pathogens, and then released into the efferent capillaries or vessels, and eventually will rejoin the circulatory system at the thoracic or the right lymphatic duct. There's a one-way flow of lymph from the interstitial fluid into the lymphatic capillaries. Overlapping endothelial cells in the lymphatic capillaries act as one-way valves and permit the entry of fluid and solutes but prevent their return into the intercellular spaces. And in point of fact, if we were to impede this lymphatic flow, we would see massive tissue swelling. Examples of conditions in which this occur are individuals that undergo surgery to remove lymph nodes because they become cancerous, or conditions such as elephantiasis, where parasitic filarial worms block these valves and impede the movement of fluid from the interstitium into the lymphatic system, causing massive swelling, edema. Lymph flows from lymphatic capillaries into lymphatic vessels towards the body's trunk. Larger lymphatic vessels contain valves that are close together. The vessels bulge at each valve. A series of bulges makes the vessels resemble a string of beads if you look at it under microscopic section. And so you can see here what that looks like. There's very low pressure in lymphatic vessels, and the valves, of course, prevent backflow. Now, the low pressure means that it's very difficult to get the lymph back to the circulatory system without some assistance. And that assistance is provided by skeletal muscle contraction and the thoracic pump. The fact that you move and you breathe helps to milk that lymph from valve to valve and eventually make its way all the way back to where the lymph rejoins the circulation at the right lymphatic and thoracic ducts as they insert their lumens into the subclavian vein on either side of the body. Where do we find lymphatic capillaries? Well, almost everywhere in the body. Areas without a blood supply have no lymphatic capillaries, and this includes the cornea of the eye. Other areas without lymphatic capillaries include red bone marrow and the central nervous system. But everywhere else you look, you're going to tend to find a lot of lymphatic tissue. If we look at extracellular fluid circulation, it flows in two different systems. It flows in the circulatory system and the lymphatic system. Blood is confined to the vessels of the cardiovascular system. It allows water and solutes to move from the plasma into the surrounding interstitial fluid. The lymph contained in the lymphatic vessels forms as the interstitial fluid drains into the lymphatic vessels and eventually finds its way back to the circulatory system after it's been filtered of pathogens. There's a continuous recirculation of interstitial fluid, which helps to eliminate local differences in the levels of nutrients, wastes, and toxins. And it also helps to maintain blood volume. It alerts the immune system to infections in peripheral tissues as a result of the macrophages and the lymphocytes that populate the lymph nodes. When we encounter a pathogen, what happens is that the macrophages will engulf the pathogen and chop them into little bits and then display some of those fragments on the membrane surface with a special protein called an MHC protein. And then any lymphocyte that recognizes that display will be stimulated to divide and form a clone army of itself that will go out and then battle the pathogen and destroy it. So here you can see the circulation of extracellular fluid. Initially what happens is that blood returns to the heart through the inferior and superior vena cava into the right atrium. That is then pumped from the right atrium to the right ventricle, from the right ventricle to the pulmonary trunk, to the pulmonary arteries which go to the lungs. And then in the lungs what happens is we oxygenate the blood, and then we return to the left atrium, enter the left ventricle, and from the left ventricle we enter the aorta, and that carries the oxygenated blood to the rest of the body. Okay? Now, what happens once that oxygenated blood gets to the tissues in the capillary beds is that we have an exchange of nutrients and waste products and oxygen and carbon dioxide, as well as fluid, from the circulatory system to the interstitial fluid. On the arterial end of the capillary, we tend to lose water and solutes, and on the venous end of the capillary, we tend to pick it back up. So you can see here that the capillaries are smallest and most delicate. In fact, they're so numerous that we don't even bother to name all of them, and they're relatively insubstantial. It's just endothelial cells and basement membrane, and that permits an ease of exchange of nutrients and waste products, as well as oxygen and carbon dioxide. All exchange between the blood and the interstitial fluid occurs here at the capillary beds. At capillary networks, blood pressure forces water and small solutes out of the blood and into the surrounding interstitial fluid. And what happens next is that about 15% of that enters the lymphatic vessels, with the remaining 85% going back into the capillary bed at the venous end, and then we re-enter the circulation. The 15% of interstitial fluid that gets into the lymphatic capillaries gets filtered, and pathogens get removed, and then we re-enter the circulation near the subclavian veins that are under each collarbone on the right and left side of the body. And so we recover the fluid that initially goes into the interstitium, and we eventually reload it all back into the circulatory system, and then the whole process repeats again. And so this is one of the hidden benefits of exercise, is that the more fluid movement you have as a result of your activity level, the more effectively you're going to filter the interstitial fluid in the lymphatic system and remove pathogens so that we don't end up succumbing to disease. Here you can see a map that shows you how lymphatic drainage works. The superficial and the deep lymphatics converge to form larger vessels known as lymphatic trunks, which empty into collecting vessels. The thoracic duct and the right lymphatic duct represent where the lymph rejoins the circulatory system. The cisterna chyli is an expanded chamber at the base of the thoracic duct and receives lymph from the inferior part of the abdomen, the pelvis, and the lower limbs through the right and left lumbar trunks and the intestinal trunk. And so you can see that lymphatic drainage is not equally distributed, right? The right lymphatic duct drains the right arm and shoulder and the right thoracic region, the right side of the head and neck, and the thoracic duct drains everything else. Now, why that is, I honestly couldn't tell you. Here you can see a little bit more detailed view of the drainage lymphatic system where it rejoins the circulatory system. Thoracic duct, remember, collects lymph from the body inferior to the diaphragm and the left side of the body superior to the diaphragm. It empties into the left subclavian vein, while the right lymphatic duct collects lymph from the right side of the body superior to the diaphragm and empties into the right subclavian vein. And so you can see those two vessels indicated here and here. Okay. Now, we talked about the fact earlier that sometimes lymphatic drainage can be impeded for a variety of reasons. Lymphedema is simply the buildup of interstitial fluid in the tissues, causing them to swell, become edematous. It's caused by blocked lymphatic drainage. The interstitial fluid accumulates and causes swollen and distended areas. It usually affects the limbs, but it can affect other areas as well. Sometimes the swelling becomes permanent because the connective tissue loses its elasticity. The stagnant interstitial fluid will accumulate toxins and pathogens, and local immune defenses will be overwhelmed, and the result often is tissue death. Now, what can we do about these blocked regions? Well, again, we talked about the fact that one of the most well-known causes of lymphedema is elephantiasis. And what happens in elephantiasis is that filarial worms block the movement of lymph from the tissues into the lymphatic capillaries. The result is that 15% of fluid we talked about doesn't get the opportunity to re-enter the lymphatic system. It accumulates in the tissues and causes conditions such as you see here. We have antiparasitics that can kill these filarial worms. One of them is known as ivermectin. And once the worms have died, the flow of fluid is restored. The result then is that slowly the tissue edema will go down and circulation will resume its normal path. But there is the problem of resecting all that loose skin and connective tissue once the condition resolves itself. Unfortunately, this treatment is only available in countries that have significant high degree of medical care, and this is one of the reasons why when we see elephantiasis, we generally pick it up in third world countries where sanitation, as we know it, isn't possible due to lack of resources. Okay, now lymphocytes are the white blood cells that are part of our immune defense, and they come in two major forms, the T lymphocytes and the B lymphocytes. They account for 20 to 40% of circulating white blood cells. Circulating lymphocytes are a tiny fraction of the total lymphocyte population. Most of the lymphocytes are stationed in lymphatic organs. Three classes are the T cells and the B cells, and a special class of T cell called a natural killer cell. All classes of lymphocytes respond to a molecular display known as an antigen. Pathogens, products of pathogens, or other foreign substances have the potential to be antigens. Most are proteins, but you can have any combination of molecules that could potentially be an antigen. You could have polysaccharide, you could have glycoprotein, you could have lipoprotein. There's a wide variety of potential antigens out there. They stimulate an immune response, and they do so because they have a surface display that's different from our own cells' antigens, and that's really how the immune system discriminates between what belongs inside the body and what represents a threat, is this presence of an unusual antigen on the surface of perhaps an infected cell, or perhaps a pathogen, or perhaps it is a non-living component, such as a toxin, that invokes an immune response. And so you can see the different classes of lymphocytes shown here. T cells make up 80% of circulating lymphocytes, and there are three major classes of T cells: cytotoxic Ts, which attack foreign cells or body cells infected by viruses. They are the primary cells involved in cell-mediated immunity. Helper T cells, which stimulate activation and function of T cells and B cells, and suppressor T cells, which inhibit the activation and function of T cells and B cells. With helper T cells, they're involved in the sensitivity of the immune response. You could think of them as the off switch to the immune response. They also are important in suppressing what we would think of as an unwarranted immune response, such as an allergic reaction. B cells make up about 15% of circulating lymphocytes, and they differentiate into plasma cells when they're stimulated in the attack against a particular pathogen. They produce and secrete antibodies, which are like little magic bullets that find a particular antigen, bind to it, and then target it for destruction by a variety of methods. They're responsible for antibody-mediated immunity, which is sometimes referred to as humoral immunity. Some B cells become memory B cells instead of plasma cells during the course of fighting an infection. They remain in reserve to fight subsequent infections of the same antigen, and the same can be said for cytotoxic T cells and memory cells. They will generate what we call memory versions of themselves, so that the next time you encounter that exact same antigen as a result of your activities of daily living, they can mount an effective attack and destroy whatever bears that particular antigen. Natural killer cells make up about 10% of circulating lymphocytes. They attack foreign cells and body cells infected with viruses, as well as cancer cells in normal tissue, and they continuously monitor peripheral tissues using a process known as immunological surveillance. So they kind of wander around the body looking for trouble, and if they find it, they destroy it. Now, one of the things that we should point out is that within these populations, the helper Ts, the cytotoxic Ts, the B cells, and not so much the natural killers, but the B cells mostly, and the T cells, you have what we call different specificities, meaning that there are a variety of possible antigens that individual clones of B and T cells can recognize. So it's not like you have one group of cytotoxic T's and B cells that can go out and hunt down all the antigens. What you have is within the entire T and B cell population a variety of different T cell receptors, B cell receptors, and antibodies that can recognize up to 1 billion different types of antigens. And the reason that this is so is because when the immune system is being created, the genes that are responsible for producing the antibodies and producing the T and B cell receptors, which help coordinate the immune response, are arranged from segments of DNA that get hooked together just like Legos. Okay, and you could imagine a Lego set with maybe 100 or 200 different blocks, and you can assemble these Lego blocks in any order you like, and that gives you a wide variety of potential things you can build with that Lego set. And the same is true for the genetic segments that make up the genes that are responsible for producing the antibodies and T cell and B cell receptors. They are assembled from segments, and the result of this assembly during the development of the immune system while you're still a newborn is up to 1 billion different specificities recognized by these T and B cell populations. So this is one of the few instances in which a body cell breaks the rule that the sequence of DNA in every chromosome in every body cell is the same from chromosome number 1 all the way down to the sex chromosomes. These guys break that rule because of the arrangement of the genes responsible for the antibodies and the T and B cell receptors. So where are lymphocytes produced? Well, they're produced in the red bone marrow and the thymus and peripheral lymphoid tissue. Different stages of development occur in each one of these compartments. The red bone marrow is important because it maintains normal lymphocyte population. Lymphopoiesis in red marrow involves hemocytoplasts that generate lymphoid stem cells. One group of lymphoid stem cells will migrate to the thymus. Remember, that's that immune organ that sits behind your sternum and in front of your heart. A second group stays in the red bone marrow and divides to produce B cells, which mature and move into lymph nodes, the spleen, and other lymphoid tissue throughout the body, such as malt and Peyer's patches and your tonsils and so on. Natural killer cells mature and migrate throughout the body and patrol peripheral tissues. In the thymus, we have a unique organ. The blood-thymus barrier isolates the stem cells that are going to produce the T cells from the general circulation, while thymic hormones, such as thymosins and thymopoietins, stimulate lymphoid stem cells to divide and produce T cells. The T cells produce and differentiate in the thymus and then re-enter the blood when they are close to being mature and travel to the red bone marrow and to peripheral tissues where they await the potential encounter with a pathogen. So it's kind of a two-stage event that leads ultimately to T cell production, and you can see a little bit of that here where lymphoid stem cells produce a more mature version of a T cell precursor, which is going to migrate to the thymus and ultimately become a mature T cell, which will then move out to the peripheral tissues. Okay, so that's what's going on with the thymus. Now, as we get older, the thymus is going to become less and less important, and the reason for that is by that time, most of the T cells have graduated T cell school in the thymus and have moved out to the peripheral tissues where they're going to do their job. And so over time, the thymus slowly begins to involute and disappear. Okay? And so you can see here the job of each of these in patrolling the body and looking for potential pathogens. Cell-mediated immunity is the job of the T cell. The B cells are for humoral immunity, and immunological surveillance is part of the natural killer cell's job. So how do lymphocytes reproduce? Well, the stem cells are found in the red bone marrow. Migrated B cells and T cells maintain the ability to make copies of themselves, provided that they are stimulated. They produce daughter cells that have the same recognition specificity, meaning they are able to bind to and destroy anything that's connected to that one particular type of antigen for which they're designed to recognize. This is crucial to the success of the immune response. Where do we find lymphoid tissue? Well, we find it in a variety of locations throughout the body. Connective tissue is dominated by lymphocytes. Lymphoid nodules are spherical masses of lymphoid tissue that are found in places like the submucosa of the large and small intestine, just to give you one example. Lymphoid organs, such as the lymph nodes, the thymus, and the spleen, are separated from surrounding tissue by fibrous connective tissue. Lymphoid nodules tend to be clusters of lymphoid cells that are found in any body system that's open to the environment. They're deep to the epithelia and they line the passages of the respiratory and the digestive system. As a group, they're called mucosa-associated lymphoid tissue. Tonsils are found at the back of the pharynx. The pharyngeal tonsil, also known as the adenoids, are located in the posterior superior wall of the nasopharynx. The palatine tonsils are located in the posterior inferior margin of the oral cavity along the boundary of the pharynx, while the lingual tonsils are deep to the epithelium that covers the base of the tongue. Occasionally what can happen is that the tonsils can become overwhelmed with pathogens, and at that point the tissue begins to become necrotic and die. And at that point it's often recommended that the tonsils be removed instead of allowing them to become a source of potential infection, and so this is what we call a tonsillectomy. Lymph nodes are small lymphoid organs that are shaped like a kidney bean. They function much like a water filter, purifying lymph before it reaches the venous circulation and removing 99% of the antigens from the lymph in the process. The immune response is stimulated as needed. When we encounter an antigen for which we are specific, the lymphocytes that are resident in the lymph nodes are stimulated to divide and make an army that will then go out and destroy anything attached to that antigen. Afferent lymphatics bring lymph into the lymph node. Lymph flows through a network of fibers and dendritic cells. It's involved in the initiation of the immune response. Regions in the lymph nodes contain B cells and plasma cells. The efferent lymphatics carry the lymph out towards the veins. So you can see here again that what's going on primarily is the checking of the body fluids for the presence of pathogens. And then if we encounter something for which we have a specificity, the lymphocytes will be mobilized to attack. The thymus is located behind the sternum and in front of the heart, and it produces hormones that are vital to the development of functional T cells. Several complementary hormones are known as thymosins. The size and the secretory ability of this lymphoid organ declines as we age, and this is correlated with an increased susceptibility to disease. But it's also a result of natural aging. The spleen is the largest mass of lymphoid tissue in the body. It has the same function for blood that the lymph nodes do for lymph. They remove abnormal red blood cells by phagocytosis. It also stores iron recycled from RBCs, and it initiates immune response by B and T cells. If we look at the ability of the immune system to defend us against disease, we have to take a sort of broad-based examination of how the body encounters pathogens and then how it deals with them. You have to remember that we have nonspecific barriers that are designed to defend us against most pathogenic attacks, and these include our skin, our mucous membranes and their secretions, and our granulocytes that are basically like crude chemical weapons that are designed to knock out anything that is perceived as foreign, but it isn't based necessarily on a particular antigen, and those include the basophils, the eosinophils, and the neutrophils, along with, of course, the macrophages. Anything that penetrates those layers is going to be subject to attack by our adaptive immunity, and that's the lymphocytes working together with the macrophages to generate a specific chemical and physical attack that will destroy the pathogen. These two forms of immunity work independently and together. Our nonspecific immunity does not distinguish one type of pathogen from another. The response is the same regardless of the identity of the invading agent. It is present at birth, and it provides our nonspecific resistance, while our adaptive immunity is what lies underneath it and has the ability, again, to generate a chemical and physical attack based on a particular type of antigen display, which could be found on the pathogen, on an infected cell, or could be a non-living disease-producing agent. There are non-living pathogens as well. Examples would be things like viruses and toxins. So our non-specific defenses include physical barriers, phagocytes, immunological surveillance, interferons, the complement system, which we'll talk about in a minute, and the inflammatory response in combination with fever. Our specific defenses protect against particular pathogens. They depend on the activities of specific lymphocytes. They produce a state of protection known as specific resistance. So here we can see some of our physical barriers at work. The integumentary system is an obvious one. Okay? It provides a major physical barrier to invasion by pathogens. There are glands that are embedded in the integumentary system that generate secretions that cut down on the ability of living pathogens to reproduce. The sebaceous and sweat glands have secretions that flush the surface. This washes away microbes and chemical agents that contain bacteria, and it also contains bactericidal chemicals that are destructive to the pathogen, as enzymes that can tear the pathogen apart, and there's also a certain amount of antibody present in these secretions as well. The hair protects against mechanical abrasion and prevents hazardous material or insects from coming in contact with the skin. And the skin itself has an epithelial layer that is multi-layered. The keratinized cells in the outer layer form a block against penetration as a result of the desmosomes that lock them together and prevent penetration. And we should also point out that it is constantly shed. So as quickly as the pathogen tries to set up camp, it will end up being shed off into the environment. Remember that 99% of dust is human skin. Epithelial barriers line the digestive, respiratory, and urinary and reproductive tracts and provide a physical barrier against attack. Their secretions contain enzymes, antibodies, or acids that can destroy pathogens. For instance, in the stomach, the gastric juice is acidic enough to destroy many pathogens before they are going to potentially enter the intestine. Mucus in the respiratory tract traps pathogens as well, and urine flushes the urinary passageways. Glandular secretions in the reproductive tract flush the tract, while mucosa-associated lymphatic tissue provides nonspecific defenses. And so you can see here an example of mucous membrane lining for instance, the respiratory system. Okay, and you can see here the mucus itself, which traps the pathogens, and you can see the mucus-producing cells down here, the goblet cells, and the cilia, which are going to propel the mucus away from the lungs and into the oral cavity, where we can either spit out the secretions containing the pathogens, or we can swallow them, in which case They should be destroyed by our stomach acid most of the time. Phagocytes are our first line of cellular defense, and they're found in peripheral tissues and remove cellular debris. They respond to invasion by foreign compounds or pathogens and can attack and remove microbes before lymphocytes detect their presence. Different types target different threats. All function in the same basic way. They essentially eat the invader, and once that happens, the phagosome, which is the invader encased in a little membrane-enclosed bubble inside the macrophage, fuses with a lysosome, which contains the hydrolytic enzymes that will destroy what's in the phagosome. That effectively kills the pathogen, and then to go one step further, we display some of those digested bits on the surface of the macrophage as a warning to the lymphocytes that there's potential infection afoot. So you could always think of the macrophages and the lymphocytes as working as a team, like a singer and a songwriter. One doesn't have a very effective way of defending the body without the other. Okay, types of phagocytes. Neutrophils are abundant, mobile, and fast-acting. They phagocytize cellular debris or invading bacteria, while eosinophils are less abundant than neutrophils, and they phagocytize compounds or pathogens that have been coated with antibodies. And we also have the monocytes that eventually become macrophages that, as we already know, coordinate the lymphocyte attack to the pathogen invasion, but also are part of our nonspecific defenses. Macrophages come from monocytes. Fixed macrophages are scattered amongst our connective tissue, while free macrophages travel all over the body. And so here you can see an example of what these different first line of cellular defense cells look like. The neutrophils have sort of pink staining granules in their cytoplasm and a multi-lobed nucleus, while the eosinophils have red staining granules in their cytoplasm and a bilobed nucleus. And then you can see here the appearance of the monocytes, the sort of kidney-shaped nucleus, the fixed macrophages, and the free macrophages being the final form of the monocyte doing its job in the body. Phagocytes can leave capillaries by squeezing between adjacent endothelial cells in a process known as diapedesis, and are generally attracted or repelled by chemicals in the surrounding fluid. So that's how they know where to go in the body. This is a phenomenon called positive and negative chemotaxis. In positive chemotaxis, we move towards the secreted chemical, and in negative chemotaxis, we move away from it. It's particularly sensitive to chemicals released by body cells or pathogens. Receptors on the plasma membrane of the phagocyte bind to the surface of the target. This is the first step in phagocytosis. After attaching, the phagocyte either destroys the target itself or promotes its destruction by activating other defenses. The inflammatory response is essentially the body's way of bringing blood to the area where there's a potential infection. And with the blood come the white blood cells, and with the white blood cells come the defense against potential infection. It's a localized response to injury in tissues and produces swelling, redness, heat, and pain. And it's caused by stimuli that kill cells and damage connective tissue fibers and or injure the tissue. The result is altered chemical composition in the interstitial fluid. Examples include prostaglandins, proteins, and potassium that gets released, as well as foreign proteins or pathogens that get introduced, and these changes trigger inflammation. And if you think about what we see in inflammation, it kind of makes sense as to why this is an effective response and also explains the symptoms of inflammation. If we think about swelling, that represents fluid coming to the area. The redness represents blood coming to the area. The heat is an effort on the part of the body to produce a temperature that is effective at promoting the reproduction of white blood cells, but it's too hot for the pathogen to survive in most cases. And the pain is simply a warning not to use the affected area while it it potentially heals. And so you can see here a flow chart showing you the genesis of an injury. Here we have a puncture wound. Chemical changes in the interstitial fluid cause mast cells to release histamine and heparin. Histamine promotes inflammation. It's generated by a special type of granulocyte called a basophil. Okay, heparin is an anticoagulant. The result here is redness, swelling, heat, and pain, and attraction of phagocytes. The blood vessels dilate, blood flow increases, and the vessel becomes more permeable, and that allows the white cells to get at the injury. Once we deal with the pathogen, we start to see the formation of a clot, which is temporary repair to protect the body from further penetration by pathogens. And the clotting is a result of a series of proteins that are always present in blood plasma that, in the presence of an injury, will generate a protein meshwork that traps platelets and other types of formed elements in blood and generates this temporary seal between the inside of your body and the outside world. Next thing that happens is that phagocytes, especially neutrophils, are attracted to the area. They pump out cytokines, and this affects immune response. Neutrophils and macrophages are in the area, removing dead cells and attacking pathogens. Fibroblasts are stimulated to come into the area and lay down new connective tissue once we've dealt with the pathogen itself. In addition, the specific defenses may be activated in order to further ensure that we don't end up succumbing to the effects of the pathogen. So if we look at our nonspecific defenses, the complement system is a group of circulating proteins that help antibodies destroy pathogens. Inflammation is that localized tissue-level response that limits the spread of injury or infection. And fever accelerates body metabolism and defense activity. Here's a look at some of our nonspecific immunity in summary, right? Fever is a rise in body temperature above 37.2 degrees centigrade, triggered by pyrogens that reset the thermostat in the hypothalamus. This is beneficial within limits. It inhibits virus and bacteria replication. It increases the metabolism of the body so we make more white blood cells, causing quicker mobilization of tissue defense and accelerated repair. Nonspecific defenses also include physical barriers that keep hazardous organisms and material outside the body, phagocytes that engulf pathogens and cellular debris, and natural killer cells that monitor peripheral tissue, releasing perforins that destroy abnormal cell membranes. Perforins are basically a protein that generates a hole in the membrane. It causes the cytoplasm to leak out and the attack cell to die. Interferons are released by lymphocytes, macrophages, or virus-infected cells and trigger the production of antiviral proteins, which stimulate macrophages and natural killer cells. It was once thought that interferons were going to be a magical antiviral and also an anti-cancer drug, and the interferons do have those effects, but it turns out that they have to be present in limited amounts, because if they're produced in high quantity, they end up destroying organs like the liver. And that was one of the things that ended up ending the interferon craze in the 80s, was the fact that many of the interferon-based drugs were deemed hepatotoxic, meaning they were bad for your liver. Specific immunity is what lies underneath our nonspecific defenses, and it protects against specific pathogens defined by a particular antigen display. Coordinated activity of T and B cells provides the specific defense. The T cells are primarily responsible for cell-mediated immunity in that they defend against abnormal cells and pathogens inside the cells, while the B cells provide antibody-mediated immunity that defends against antigens and pathogens in body fluids. And the difference between these two is that the T cells have to be present right at the location of the pathogen or the infected cell to be effective, while the B cell can be effective at quite some distance. from the pathogen because the antibodies travel in the plasma until they find their antigen that they recognize and then trigger the attack. So there's two types of specific immunity. Active immunity occurs when the body develops antibodies in response to a particular antigen, while passive immunity occurs when we receive antibodies from another source. Both can be naturally acquired or artificially acquired. So let's deal with artificial immunity first, okay? Let's say that you were traveling to a part of the world where malaria was an endemic disease and you wanted to be essentially protected against the malarial parasite. You could get an injection of antibody from a different source that's directed against the malarial parasite, and that would provide temporary immunity for a period of time until the antibodies cleared from your system, usually about 2 weeks or so. Passive natural immunity would be a similar state in which you get antibodies from another source. Two examples would be the mother passing antibodies through the placenta to the baby to protect it against pathogens during development, and also the baby picking up antibodies from breast milk as it nurses, first on colostrum and then on breast milk during its neonatal period until the baby's immune system is competent to defend against pathogenic attack. In active immunity, what happens is that you're exposed to an antigen that is unlike your own, and you generate an army of lymphocytes that are going to protect you against that encounter with the antigen initially, and then provide memory of that particular defense against invasion for a subsequent exposure. And so in artificial active immunity, you get an injection of antigen harvested from the pathogen. The result is that you generate B and T cells that will provide protection against that antigen. And in turn, if the actual pathogen shows up bearing that antigen, you are safe. And in natural active immunity, you encounter the living pathogen, generate an immune response against it, knock it back, and then the next time that same pathogen shows up, you're immune. So the important thing to understand about active immunity is it's lifelong, and passive immunity is temporary. If you think in terms of exposure to pathogens in your environment, basically the more exposure you have, the more memory cells you build up, the more antigens you can defend yourself against, the less likely you are to become sick in the future. And so that's part of what makes our specific or adaptive immunity so effective, is this memory function. So this is one of the reasons why frequently if you were very sick as a child, you tend to be relatively healthy as an adult, in part due to this massive army of memory cells that you've built up, that in the event we have a second encounter with the pathogen, will rapidly produce B and T cells designed to destroy this subsequent invasion. Now, very important point is that the pathogen has to make a living too. And this is one of the reasons why you might come down with the same illness maybe several times in your life, but you're not succumbing to the same strain of pathogen when that happens. What's happening is that the pathogen is mutating. As a result, it's changing its appearance, and that allows it to enter the body and produce an infection. And so you have to start the whole process of the immune system recognizing the pathogen all over again at that point. So the properties of specific immunity include the ability to recognize a particular antigen. We call this specificity. Each T or B cell has receptors that bind to one specific antigen but ignore the others. The response of activated T or B cells to a specific antigen is a characteristic that is endemic to one particular T or B cell clone. Okay, remember we talked about the fact that these specificities are generated as the immune system develops inside you, and it results in individual T and B cells that are generated as the immune system begins to form um, having their antibody genes and their T and B cell receptor genes arranged in such a way that they're designed to recognize and attack only a particular antigen. This is good because if we were to stimulate every lymphocyte in the body every time you had an infection, we wouldn't effectively defend you against invasion because we would be clogging the system with lymphocytes that are not designed to attack and destroy that particular pathogen bearing that antigen. It's also versatile. Millions of different lymphocyte populations are sensitive to different antigens, and when activated, the appropriate lymphocyte divides, producing more of the same lymphocyte type. That's what we call creating an army of clones. It's also got memory, okay? Activated lymphocytes provide two groups of cells, a group that attacks the invader immediately, and a group that remains active unless exposed to the same antigen at a later point. Memory cells that enable the immune system to remember an antigen and launch a faster, stronger, and long-lasting counterattack in a second exposure is an important part of our adaptive immunity. It's also tolerant of our body's own antigens. It ignores the antigens present on our body cells, but only targets those that are not present. It can develop over time in response to chronic exposure to an antigen. You can also, over time, tolerate certain foreign antigens if you're exposed over and over and over again. Now, sometimes the tolerance breaks down, okay, and the immune system attacks the body's own tissues, and we call that autoimmunity. Okay, so let's take a look at your specific defenses and what triggers them. Exposure to an antigen will activate phagocytes, and they will internalize the invader and chop it into little bits and present those little bits on the membrane surface along with a protein called a major histocompatibility protein. It stimulates cell-mediated defenses involving attacks by T cells and antibody-mediated defenses, and that results then in an army of B and T lymphocytes that only respond to that particular antigen, whether it be on an infected cell or on the pathogen itself. So the first step is antigen presentation. So you can see here sort of an overview of the entire process. Okay, the invader comes in, the phagocytes encounter it, and they stimulate T cells by direct interaction. In addition, T cells also communicate with B cells that are specific for that particular antigen as well, having been sensitized by an interaction with that particular antigen early on. The cytotoxic T cells engage in direct physical and chemical attack on infected tissue or the pathogen itself. The activated T cells find the pathogens and attack them through phagocytosis and the release of chemical toxins, while the B cells, once stimulated, will produce massive amounts of antibody that will flow through the blood plasma and eventually find their antigen target. And when they attach to it, they'll promote a variety of methods to render anything bearing the antigen as ineffective at producing disease. Examples include cloaking the antigen if it were a toxin and rendering it ineffective. Another example is stimulating the complement cascade, in which plasma proteins that are designed to ultimately lyse an infected cell or pathogen are triggered to activate by interacting with antibody that is bound to antigen on an infected cell or on a pathogen. Another way is through agglutination, where we take the actual pathogen, and by connecting the antigens of one pathogen to another using the antibody like links in a chain, we immobilize the pathogen so that it can no longer produce disease. And then it also aids in a process known as opsonization, where we attract macrophages to infected cells or to pathogens that are coated with antibody, promoting either a chemical attack that destroys the infected cell, or in the case of a pathogen that's coated by antibody, it makes it easier to engulf it and destroy it. Antigen-presenting cells are specialized cells. Examples include monocytes, macrophages, and dendritic cells. They engulf a pathogen, digest it, and produce tiny fragments that are presented on the membrane surface in combination with a major histocompatibility protein, and that serves as a signal to stimulate the appropriate B or T cell to generate a clone of itself. Again, it's this combination of molecules on the surface that is the trigger for one particular flavor of B or T cell, and not every B or T cell at large. The defenses against the pathogens include activating macrophages, antigen presentation by macrophages or by infected cells serve as a way to attract the immune system to those cells. Defenses against viruses involve direct contact with virus-infected cells and antigen presentation by APCs. Essentially, this is a way of telling the immune system that the pathogen is present. Antigen-presenting cells include things like macrophages and dendritic cells and so on. But we should also note that infected cells have the ability to present antigen as a way of indicating that they're in trouble. And in this case, the signal is, instead of stimulating the clone cells to divide, the primary purpose of the signal is to attract lymphocytes to the area so that we can destroy the cell that's presenting that antigen. The release of interferons by infected cells also warns neighboring cells that there's a pathogen in the area, and they generate molecules in their cytoplasm that interfere with viral replication. So you can see here how the interferons do their trick, as well as the antigen-presenting cells. And here we can see how the macrophages are operating. Okay. Memory cells have no role in the initial infection. They are generated during the course of defending against the invasion. They dramatically reduce the response time for subsequent infection by the same pathogen. Upon secondary exposure, they differentiate into cytotoxic T cells, helper T cells, and plasma cells. Okay, so what you have here is an entire suite of lymphocytes that have already been selected for their ability to recognize that pathogen, and when we have a secondary exposure, they're able to produce such large numbers of defense cells that the pathogen doesn't get a chance to make you sick. This is a rapid and very effective response against disease. An antibody is basically a protein that's designed to attach to a particular antigen. It's made up of two parallel pairs of polypeptide chains, one pair of heavy chains and one pair of light chains. Each chain has a constant and a variable region. It's the variable region that actually attaches to the antigen, and the constant region is the same across all classes of antibody. The constant region form the base of the molecule, and they indicate the class of antibodies. Some antibodies are designed to float free in blood plasma, for instance. Some are designed to be attached to tissue inside lymphatic organs and not to move. We call those antibodies that are fixed. And some are designed to be membrane receptors. The antigen binding sites are the free tips of the two variable segments, and you can see those here. Let's circle those in black. Okay, so on this particular model of an antibody, this would be an antigen binding region, and this would be an antigen binding region. Okay, so this is really the business end of the molecule. The rest of the molecule, the constant region, is going to be the same regardless of what antibody we're talking about. So there's a framework that serves as a basis for attaching the antigen binding site to the entire molecule that's held in common by every antibody. These tips can interact with antigens similar to the way that an enzyme interacts with a substrate, and it's the differences in the variable segments that affect the shape of the binding site and determine its specificity. Remember that antibodies are going to be specific for different types of antigens. Variation occurs during the production, division, and the differentiation of B cells. And it's the result, again, of a variety of methods to generate these genes that are responsible for producing, in this case, the antibodies, as well as the T cell receptors. So if we were to kind of sketch what goes on here, remember our Lego example, right, where we actually assemble the antibody from DNA fragments that are scattered throughout the genetic material of one particular chromosome, and we can join these fragments in any order to create the specificity for the enzyme. So let's say, some of these fragments will indicate as having a particular sequence by coloring them different colors. Okay, so we'll generate now a variety of potential. This is just a tiny section of, say, all the potential DNA segments that could contribute to the gene that codes for the antibody. Right, so here we go here. And then here we go down here. Okay, so this would be in the DNA of the developing lymphocyte, right? And so you have to think that there's a wide variety of these little DNA fragments. And when we generate now a novel gene, what happens is that we combine those fragments in such a way that we're able to generate now a functioning gene. So let's say in our example here that we were to hook up a certain set of fragments, let's say the green and the blue and the white fragment, right? So there's our blue fragment there, our white fragment here, and then our green fragment over here. Right? And that might code for, let's say, the light chain of a particular antibody. Okay, so this would go on in the DNA and produce a transcript, mRNA transcript, which would produce now a particular light chain of antibody. Let's make that, I don't know, gray here. Okay, so there's our light chain. Okay, and then you can imagine that this light chain can combine with any number of different heavy chains, and so you can see that by mixing and matching the segments, and by mixing and matching the light chains and the heavy chains, you have the potential to build almost any type of defense molecule that's going to keep you safe from an invading pathogen that contains an antigen other than your own. Okay? And this all goes on, again, while you're developing in your mother's womb and during the first few months of neonatal life. Okay, we're producing now these ready for prime time B cells and T cells. Right? That's how we get these billion different specificities. The constant region is not something that would be assembled from these diverse bits of DNA that are in the let's connect these so that you can understand they're all on one DNA molecule in a chromosome there. Okay. The constant region would be the same block of information regardless of the DNA that codes for the antigen binding region. And again, the constant region determines the antibody's class, okay, whether it's free floating, whether it's membrane bound, whether it's going to be stationary inside of a lymphatic organ, for instance. The antibody binds to the antigen to form an antigen-antibody complex. The binding between the antibody and the antigenic determinant is the basis for the attack of the immune system against the pathogen that bears that antigen. Specific portions of the exposed antigen surface represent the binding site for the antibody, and a bacteria may contain millions of different antigenic determinants, and as a result could be attacked by a variety of different B and T cell clones. Complete antigen has at least two antigenic determinant sites, while partial antigen, or hapten, does not normally activate a B cell. It has to be present in combination with what we call a carrier molecule in order for the antibody or the T cell receptor to recognize it. It can attach to the carrier molecule and function as a complete antigen in that context. The antibodies will attack the hapten and the carrier molecule, and ultimately this will lead to its destruction. If the carrier molecule is normally found in tissues, then the antibody could attack normal cells, and this is thought to be the basis for drug reactions like allergies to penicillin. Okay, so the penicillin on its own is not problematic, but when it interacts with normal body molecules, that's when things become a problem. So these are some of the different antibody classes. There are 5 classes of immunoglobulins that are determined by differences in their constant regions, specifically the constant regions of the heavy chain segments: IgG, IgE, IgD, IgM, and IgA. Okay, so what do we mean by different classes? Well, they have slightly different areas where they work. Okay, we start with IgG. These are antibodies that are responsible for resistance against viruses, bacteria, and their toxins, and they account for 80% of antibodies. IgEs attach to basophils and mast cells, causing them to release histamine and promote inflammation. Okay, so see how the constant region down here is a little bit different than it is in the IgG. IgD is an individual molecule on the surface of a B cell, so it acts as a B cell receptor where it can bind antigens in the extracellular fluid, and this binding plays a role in sensitizing the B cell and allowing it to become activated, usually with the assistance of a helper T cell. So you get sensitized first, and then the helper T cell, which is responding now again to that particular antigen, having encountered it on an APC, will secrete chemicals that will promote the rapid cell division of that particular B cell clone. IgM is the first class of antibody secreted after an antigen is encountered. IgM concentration declines as IgG production over here picks up. The anti-A and anti-B antibodies responsible for the agglutination of red blood cells are IgM type antibodies. And these are generally not as effective at promoting inflammation as the IgG antibodies. And as a result, this is one of the strategies employed in giving people what we call allergy shots or desensitization therapy. The idea is that we inject them with a particular antigen that proves to be their allergy trigger, and they increase the amount of IgM circulating in the plasma, and then when they come across small amounts of antigen as a result of their activities of daily living, we don't promote the inflammatory response as effectively because the IgMs are busy picking off all of this presented antigen, and as a result we don't get the histamine release that is part and parcel of interaction with things like IgE, right, where we kick those basophils up. IgA is found primarily in glandular secretions like mucus and tears and saliva and semen. These antibodies attack pathogens before they gain access to internal tissues. And so you can see here, this is what we call a monomer. These are all monomers. This is a pentamer, five-sided shape, right? 5 separate subunits all attached to each other. And over here, this is a dimer. Okay, there's one antibody there. There's the other down here. So these are different classes. This only speaks to their role in defense. This doesn't speak to their specificity for the antigen. You can have the billion different sensitivities present in molecules of IgA, IgM, IgD, IgE, IgG produced by the appropriate lymphocyte. Okay, this is just a look at the difference between the primary and secondary response to a particular antigen on a pathogen. In the primary response to antigen exposure, it takes time to develop. The antigen activates the B cells, which differentiate into plasma cells and secrete antibodies into the plasma. The antibody titer peaks 1 to 2 weeks after exposure and declines if it is no longer exposed to the antigen. You can see here with the primary response that the first antibody to kick up is the IgM, and then after about 2 weeks pass, we peak with the IgG as the IgM levels decline. In the secondary response, it looks quite a bit different. What happens is we have a second exposure to the same antigen, and remember now we've got a waiting army of lymphocytes that have the appropriate specificity to battle that particular pathogen bearing that antigen. The result is that we still see an IgM response, but the IgG response is much more dramatic. Kicks up in under a week. It lasts longer than the primary antibody titer. Increases more rapidly. The result is immediate response by memory B cells that appears even if a second exposure after the first exposure is 20 years or more down the road. And so this is one of the reasons why the adaptive immunity is so effective at keeping us disease-free the majority of the time. Now that said, it's not a perfect system. We've talked about the fact that there are hypersensitivity diseases, allergies for instance, which are basically overly vigorous immune responses to an antigen that doesn't really present a threat to the body. And that's primarily due to an initial exposure that sensitizes mast cells, and then with the subsequent exposure we see massive histamine release and inflammation typical of what we note in allergies, and so you get the watery eyes, the itchy skin, the runny nose, and so on. And then we have autoimmune disorders where the immune system, which normally tolerates your body's own antigens, turns and destroys cells that it normally leaves alone. And the mechanism behind that is a little bit of a mystery, but we think that there's maybe a few possible explanations for why this would occur. One of these would be a mutation in either the immune system or the body's own tissues. If it occurs in the immune system, we could produce a T cell receptor or an antibody that we didn't initially produce when the immune system was being formed, and it could end up being a bad actor attacking our body's own tissues. Another possibility is a mutation in the tissues themselves, changing their antigen presentation, and again, then revealing them as a target for attack. Another possibility is breaking the barrier between tissues that have immune privilege and those and the blood supply and the immune system. So there are some areas in the body where the immune system is basically locked out and can't get in. Those include things like the cornea of your eye, your central nervous system, and in males, um, the uh, the duct system that provides the route for sperm to get to the outside world, which includes the seminiferous tubules, the epididymis, the vas deferens, the urethra. And in females, we kind of do the same thing by cloaking the female's eggs in body cells to disguise it from the immune system. Those are called follicle cells, and the cloak they form is called the corona radiata. And then your liver also has immune privilege. And if we break the biological barriers that provide that, the immune system can come in and wreak havoc. For instance, in men that get vasectomies, where we break the blood testis barrier, we generate antibodies against our own sperm cells, and that makes it very difficult to reproduce now because we've exposed the immune system to something that it hadn't seen before, your own gametes, and the result is that they will mount an attack against those cells. Another possibility, and probably these two are probably more common than any of the previous three that we've discussed. One is exposure to a cross-reacting antigen, and so if we like, let's sketch an antigen, let's say that's on our body's own cells, and let's say that this is a cell surface, right? And this is our antigen. It has this kind of shape on it. And so we don't generate an antibody that recognizes this antigen because it would cause an attack on our own tissues. But let's say that we get exposed to an antigen that is similar but not exactly the same as our body's own antigens. And let's say it's on like a pollen grain or something. So there's our little pollen grain. And then we generate antibody that recognizes this, okay, and attacks and destroys it very effectively. Right? There's our heavy and light chains. Now this guy gets wiped out, but you've got all this antibody hanging out. And you've also got large numbers of B and T cells that recognize this antigen, which is similar to but not identical to our own. And so the feeling is that in autoimmune diseases, this can result in cross-reactivity against our body's own antigens, and that produces the manifestations of the disease. Another possibility is that B or T cells that have been rendered in a state of anergy, meaning they're alive but they're not actively participating in the immune system, get turned back on somehow. And they would be the cells that had been rendered incapable of being part of a functioning immune system because they would result in autoimmunity. Well, these guys get activated, and then they go out and attack the body's tissues. Okay. Could be a problem with the regulatory T cells. Could be a problem within the cells that have been rendered in anergy on their own. We're not really sure how and why this happens. So what are some diseases that fall under this category? Well, rheumatoid arthritis is one. Okay. Type 1 diabetes is another. Lupus is another. Lupus erythematosus. Okay. Graves' disease. Okay. These are all examples of autoimmune disorders. Okay. What's the treatment? Well, the treatment is to knock down the immune system by giving you anti-inflammatory drugs and immune suppressants. And the problem, of course, with this is that it leaves you open to infection. because you're battling your own immune system. And we can't yet pick out just a particular group of lymphocytes from all your lymphocytes to turn down or to turn off or even maybe to kill. We don't have drugs that are capable of doing that yet. So we sort of take this blanket shotgun approach that reduces your overall immunity. The same thing is true with allergic reactions. The bad actor here is the immune system, and so we give you anti-inflammatories as a way of controlling the agent responsible for your discomfort, which in this case is your immunity. Okay? So, hopefully this was informative. I will see everybody in the next podcast. Remember to let me know if you have any questions. Thanks for joining me.